If they are not able to be treated with these drugs, there's very little that ultimately can be done, and so there's ultimately a very high mortality rate, and that's the greatest concern. A drug-resistant bacteria is a bacteria, typically it's a pathogen that we're concerned about, so a bacterial pathogen that has, through a variety of mechanisms, developed an ability to avoid or degrade a drug so that the drug's no longer effective in killing that bacteria. So it's in the news because there's a particular grouping of these bacteria that have developed resistance towards a common drug that we would use in the clinic, carbapenem. And they do so through a mutation that has developed a particular enzyme in these bacteria called beta-lactamase. And these enzymes then are very effective in degrading the drugs that we would typically use. So we could treat these bacteria with these drugs, but this enzyme then breaks them down so the drug is no longer effective in killing that bacteria. It means that then physicians have very few options to treat patients. And so if a patient has a sort of severe infection, then we've really run out of tools. If that infection is not responsive to any drug that we would potentially use, then we've really run out of tools to treat that patient. Those have been phenomenal drugs, and we've now come to a scenario where we've overused them. And we've promoted then the emergence of these drug-resistant bacteria that are now becoming more difficult to treat because we're running out of drugs to treat them. So it's important that we both use the resources we have effectively and carefully, and then to complement that with the discovery of new compounds that will be effective against these drug-resistant bacteria. So in my group, we're really interested in developing new therapies that then are going to utilize mechanisms that current antibiotics do not. And so the type of compounds that we're developing are small fragments of proteins known as peptides. And so we're developing peptides that specifically kill bacterial pathogens and do so through mechanisms that current drugs are not employing. And so, these compounds then can be effective against bacteria that have become resistant to common antibiotics because, again, they're hitting targets in the bacteria that current antibiotics are not. So it's just a matter of time that the drugs that we have will ultimately become not as useful as they once were, and so it's really important that we stay ahead of that in this sort of arms race against the bacteria and developing new compounds and funding work to discover those new drugs that then will be the next antibiotic that we could potentially use to stay ahead of this resistance that these bacteria are developing.
Yeah.
Okay, so you just have to make sure you have a move on, just like, those sort of like background footage and everything. Use our
Yeah. We still have something rough left over.
Okay.
Just always start with liquid culture.
Fluorescence is much better. So typically it would be like antibiotics.
We were gonna do
and then, if that goes well, I don't know what the next step would be. Maybe they will then